All right, let's move on to lecture 14, cholesterol metabolism and membrane. So this is going to be much more of a generic after we get through the metabolism stuff. So let's get into it. So kind of what I was alluding to in the previous lecture, which was, these isoprenes being like the building blocks for sterols and steroids and cholesterol and all that. Let's move on to how it's actually done, how this mechanism works. So the first committed step is the synthesis of mevalonate, which is just a 6-carbon molecule. So this is our committed step. So combining 3 acetates, again, acetate means that it's a 2-carbon molecule, combining 3 of them will form our 6-carbon mevalonate, and that's our committed step. And it uses 2 NADPHs to do this whole committed step and to form our 6-mevalonate. Now, the second step is our activation, where we use 3 ATPs and we form activated isoprenes. So we basically take the 6-carbon and get rid of one of the carbons to form the 5, and then we add 2 phosphate groups essentially, and then another ATP is used in the process as well. And these are things you have to what's it called, memorize, but definitely do know this whole pathway. And it's going to release CO2 and the inorganic phosphate. And then number 3 is condensation into squalene. So squalene is just a bunch of these isoprene monomers coming together. So right here, 1, 2, 3, 4, 5, and then it's 1, 2, 3, 4, 5, 1, 2, 3, 4, 5, and so on and so forth. We just combine a bunch of them. And so squalene is our 30-carbon molecule, so that means we need to use 6 of these isoprenes. So right here we have 1, 2, 3, 4, 5, and 6. We have 6 isoprenes coming together, and that ends up forming our 30-carbon unit. And so the steps are, we add 5 plus 5 to form 10. We did this twice, and now we have, the way we do this is 5 plus 5 is 10, 10 plus 5, 15, 15 plus 15 is it forms into 30. So we don't just keep adding 5 the entire time. It ends up just combining 2 of the 15s together. And this third step uses NADPH. And then the last one is cyclization into cholesterol, which we form our 4 fused rings. And we use NADPH and oxygen. And the oxygen will basically be converted into water. And something I want to note is this oxidosqualene cyclase. Remember, that third step where we form our squalene, we end up using NADPH. And so it means it's an oxidation reduction reaction. That's what this oxido part talks about. So this enzyme has 2 properties. It catalyzes cyclization reactions. This is step 4. That's my apologies. So for step 4, it's going to use the cyclase activity and to form into a cycle. But also, it's going to do the membrane insertion of the product. So spatially, it'll shuttle it into the membrane, the nonpolar region, and traversing this polar region. So that's what you need to know about that in terms of that specific enzyme. And then cholesterol fates and regulation. So some of the fates is that when you develop the cholesterol, it can be turned into cholesterol ester, which we know goes into the blood essentially because that adds an OH group and makes it polar. And then it could also be turned into many hormones, progesterone, aldosterone, testosterone, cortisol, estrogen, yada, so on and so forth. But it's important to balance between synthesis and dietary uptake. And so the major point of regulation is HMG-CoA reductase, right? So HMG-CoA reductase, if you look at this diagram right here on the bottom right, we see that it catalyzes the reaction between beta-hydroxy, beta-methyl-glutarate-CoA, essentially I think it's a 4-carbon, if I'm not mistaken, into mevalonate, right? And so what's going to end up happening, I'm pretty sure. Yeah, it's an intermediate. But this HMG-CoA reductase is the enzyme that can convert it into mevalonate. So this is one of the enzymes in the committed step, and that's step 1 for cholesterol synthesis, and it's stimulated by insulin, and it is inhibited by low ATP, glucagon, and oxysterol, which is just a cholesterol metabolite. So if cholesterol breaks down, then obviously we don't need more cholesterol. And HMG-CoA reductase is the target of many cholesterol-lowering drugs because we're stopping the body from producing its own, and it's often just in competitive inhibitors, so it'll competitively bind to this HMG-CoA reductase with beta-hydroxy, this CoA thing. I don't want to say the whole name. And they're often called statins, which I'm sure you guys have heard before. And why target this enzyme and not others downstream? And that's just because that the steps afterwards require ATP and NADPH, so they're already not favorable at all. So now we're moving on to more of the lipids, energetics, membranes. This is more of like an overview. This should be a little bit easier to go through. So energetics of lipids. So there are different ways that these lipids can react when exposed to water. So dispersion of lipids in water. And what ends up happening is the water will form a shell around the fatty acid. And this actually causes the entropy to be decreased. And we know that the universe wants to increase in entropy, right? And so it's unfavorable because nature always pushes towards more disordered, meaning that the entropy is increasing, like I just mentioned. And so then the next thing is clusters of lipid molecules. So as you can see here, here's the clusters of lipid molecules where we essentially have these lipids forming like a little like non-polar region, and then you have the polar water shell like around it, essentially. And so the lipids is called lipid aggregation, where the outer lipids make the water disordered, the middle lipids don't affect the entropy of water. And so if you look at the bottom left versus the top right, the aggregation causes less water molecules to be ordered. And so it's going to favor this, because if we have the lipid already introduced into a water solution, this is going to be super unstable. This is unstable, but it's a little bit more like disordered, if that makes sense. So the universe wants that. And then the last step is micelles, which is, you can think of them like vesicles, but they're not exactly the same. Vesicles have this inner layer as well, like an inner cavity that has another hydrophobic or hydrophilic heads, phosphate heads that are on the inside. And I'll show you an example in a few slides. And so the ideal scenario where hydrophilic heads are facing the water, forming basically making the water as the highest level of disorder as possible, and then it allows for water to be less ordered, and then it allows for the hydrophobic tails to face inward and away from the water itself. And so this is what I wanted to emphasize is like there's a micelle, bilayer, and vesicles. So in micelles, they're just basically like soap molecules. The individual units are wedge shaped. So each phosphate tail has this like wedge shape. You should definitely memorize this. Then the phospholipid bilayer is a cylinder shape where we have two fatty acid tails. Whereas in the micelle, we only have one fatty acid tail. Something to know as to why they have these shapes is the one fatty acid tail has a wedge shape, whereas the phospholipid has two fatty acid tails, which allows it to be cylinder shaped. And that's a bilayer, which we all know what that looks like. It's canonical. And then for the vesicles, I was talking about this aqueous cavity, and there's this other layer of hydrophilic heads along with the outside, and then you have this hydrophobic layer in the middle. And so that's what a vesicle is, and that's what's the difference between a micelle and a vesicle. And if we had to predict what is the shape of the individual units in a vesicle, they're also cylinder as well, because again, each phospholipid head or hydrophilic head has two fatty acid tails attached to it. So it makes that cylinder shape. So now cholesterol effects on the membrane. So cholesterol are membrane plasticizers, and they interfere with the motion of fatty acid side chains. So essentially, if we have an increased high temperature, right? This is what from this state to this is doing. This is high temperature, this is like a lower. And so this high temperature causes the fatty acids to move a lot in vibration and cause disorder, but we know that they want to stay together, right? Like a lot of these want to like flip around and everything because they have high energy, but they don't want to in terms of non-covalent interactions with the water. They don't want to flip, but you know, the environment's kind of making them and forcing them to. What cholesterol does is it inserts itself and increases like the TM, or essentially the ability of this lipid to resist high changes of temperature. That's essentially what happens. And so again, it lessens the abruptness of transition, so high temperatures, and membrane starts to melt, like as I mentioned. And the cholesterol, the way it does it by mechanism of action is it raises the melting point of the membrane, and the addition of unsaturated phospholipids will come in and basically have a fluid, liquid, distorted state, which just means that when it's high temperatures, we're going to have a higher concentration of unsaturated phospholipids that are integrated into the plasma membrane. And then when membranes start to freeze, temperature below freezing point, the cholesterol prevents ordered packing because it fits itself in. So don't try to connect the two, because I know it's very tempting to say, well, if cholesterol does this in hot, then why doesn't it not do the same effect in the cold? It's because they're different. So just think of them differently. In the hot, cholesterol behaves this way. In the cold, it behaves this other way. And that's the best way to keep things straight in your head. And then talking about the fluid mosaic model, basically a lot of proteins, different kinds of proteins, channels, glycoproteins, all this stuff, signaling, transport. There's a bunch of them in here, and it's integrated randomly throughout the membrane. It looks like a fluid mosaic, like the Van Gogh Starry Night. I think that's what I always think of when I hear fluid mosaic. But anyways, that's like a little side tangent. But yeah, so different compositions for different structures. As you can see, plasma membrane has cholesterol, sphingolipids, phosphatidylcholine, phosphatidylethylamine. These are mouthful, and then minor like lipids, and then that's going to change in comparison to these other membranes that are in the organelles or whatnot inside the cell. And so talking more so about lipid transfer, there are these things called lipid transfer proteins where lipids can move between membranes via these types of proteins. And there are three kinds. There's monomeric, which is a single lipid transfer protein. There is oligomeric, which is just a few multiple lipid transfer proteins that move it and they're just connected together. Or there's ATP driven, which uses ATP to drive the lipid transfer itself. So those are three. And then membrane dynamics. So we have three types of diffusion. We have lateral diffusion, which just happens on its own. Obviously, it moves within the layer itself. The phospholipid layer isn't just static, it moves around. That's the first one. Then we have flip-flop, where we don't use necessarily a catalyst, and it just flips from one end to the other, and it does so spontaneously. And then we have catalyzed trans-bilayer translocation, which all that means is like we're flipping them, but we're using an enzyme to help and to kind of regulate which direction is going to be the most favorable, you know. And so flipase, and these are annoying names, but flipase is going to move our phospholipid tail from the outer leaflet to the inner leaflet. And then flopase is going to do the opposite, where it moves it from the inner leaflet to the outer. And then scramblase, it's just a random direction. It'll either go from inner to out or outer to in. And it just does so randomly in an equilibrium. So it's almost like these are unidirectional, the flopase and flipase. They are unidirectional. And then scramblase serves to like equilibrate everything. So that's a very good thing to know about these three different types of catalyzed transbilayer location. And then this is something called FRAB, which is fluorescence recovery after photobleaching. And the reason why I bring this up is because this is how we figured out this lateral diffusion right here, where if we had a sheet of plasma membrane, the lipid can move laterally in any direction, x, y. Not necessarily the z, because z would mean it would flip, but the x, y direction, it can move any direction. And so we found out because what we did is we introduced fluorescence we basically added antibodies that were bound to the hydrophilic heads of the plasma membrane. So when we're looking at it from a top bird's eye view, we see a bunch of red dots, right? What we do is we're inserting a laser beam that bleaches this area. So meaning that we basically destroy these antibodies and they no longer fluoresce. And so as you can see here, it's bleached. There's no fluorescence. There's no red signal here. And what ends up happening with time, when we keep taking photos progressively from time 0 to whatever, we see that the red starts to occupy this area spontaneously. It's not like the antibodies turn back on. It's the fact that they diffuse laterally. So that's the pretty cool, remarkable thing about it. And then membrane proteins, there's integral, peripheral, and amphitrophic. So integral is like they're firmly associated, as in like this right here, where they basically span both the membranes, the inner and outer leaflet, if we're trying to be specific. And then the peripheral, they're typically they're only in one leaflet itself. And they're usually associated with the membrane via electrostatic interactions and hydrogen bonding, whereas integral proteins are associated with the membrane via hydrophobic interactions, which are much stronger in this case within the. And then amphitrophic are proteins associated reversibly with the membrane. So if I can find one, it's like right here, where they can bind, unbind, and do so in a reversible equilibrium reaction rate. And same thing with right here, PIP. And then tyrosine trip residues are common at the water lipid surface interfaces because they have the polar region and then they have the non-polar parts of like the rings and everything. And then some membrane proteins have beta barrel structures, which allow for things to pass through or not pass through when they serve as signaling, a bunch of stuff. They just know that there's beta barrels. And then hydropathy plots are essentially different ways to categorize hydrophobic versus hydrophilic. And so in the y-axis, you have hydropathy index, which it starts from 0 and it moves in both directions. The positive direction means that you are hydrophobic because it's a hydropathy. So we're measuring the hydrophobicity with the positive values. And then the negative values are going to be the hydrophilic, which are going inward. And then the x-axis will tell you the residue number, right? And the way you tell the residue number is by these large humps, right? And if these weren't color-coded, you can tell these are a larger hump than these bottom ones. And you can tell that in the top part, it's hydrophobic, right? And so what that means is that for number 1, this is the first transmembrane segment that is inside the bilayer, right? It's in the hydrophobic region. And this is in the hydrophobic region. And 3, 4, 5 are in the hydrophobic, 6, and 7. And there's 7 separate subunits. If you're ever given a hydropathic plot and they tell you how many segments are inside the phospholipid bilayer, it'll be much more clear than this because you can argue 6 and 7 are the same one. But it'll be much more clear than what is on here now. And then number of hydrophobic regions equals the number of times the protein crosses the membrane. So in this example, the protein crosses the membrane 7 times because in order to cross membrane, you have these alpha helices that spam that hydrophobic region, and that's exactly what's here in the 1, 2, 3, 4, 5, 6, 7. So good thing to know. And this might be a GPCR, you'd have to guess, because GPCRs contain 7 transmembrane domains. That's something to know in terms of how to interpret from these transmembrane plots. I know that, for example, voltage-gated sodium channels have 6 transmembrane segments, so you would see 6 different humps on there, essentially. And then in terms of membrane fusion, that's the next part. So there are different types of membrane fusions. There's exocytosis. You expel stuff outside from the cytosol into the extracellular matrix. You have endocytosis, which is the opposite direction. Then you have viral infection, which we don't even need to go into it. Then we have fertilization with the sperm and an egg. And then we have cell division, which separates the two types of cells in mitosis. Membrane fusion during neurotransmitter release. So basically vesicles containing neurotransmitters contain V snares. So this is a form of exocytosis, but it's using vesicles. So with neurons, they have vesicles that need to be almost exocytosed, like the neurotransmitters inside, exocytosed immediately. And the way they facilitate this is using these proteins called snares. And all it does is it coordinates it, and almost imagine like it's forcing the fusion. It's bringing together the vesicle and the plasma membrane and combining them so that the neurotransmitters are pushed across the membrane and into the extracellular space. That was very complicated. We'll go through this one by one. So the V snare is a protein on the vesicle, hence the V. Whereas T snare, the T is corresponding to the transmembrane, plasma membrane. I don't know. That T is for the plasma membrane, essentially. I know it's like it should be a P, but it is what it is. And so these two are going to come together and almost like form cross bridges and like connect. And so as you can see here, they're basically connected and they're tethered together. Now what's happening is like you pull it across and then the two things are forced together, right? The vesicle and the plasma membrane are forced together. And then what ends up happening is the plasma membrane will split in half and then the neurotransmitters can now exit out. And this is just a form of complete fusion where the pore widens, thus releasing all the neurotransmitter. And that's the way we have exocytosis in neurons. Right, I want to emphasize this is called hemifusion, where the inner leaflets of both membranes come into contact, favored because the zipping causes curvature and lateral tension on the bilayers. And the models for protein-induced curvature. So there's a couple models that allow for the membrane to be curved. The first one is protein with intrinsic curvature that allows it to interact with some of the polar groups on the hydrophilic heads. The second one is a protein with one or several amphipathic helices embedded into one leaflet that force the membrane to bend. So it's almost like they get a grip on the plasma membrane and then they squeeze it together to form that bend, right? And the last one is proteins with bar domains that polymerize into superstructures that force the membrane to bend. And this is, you just have to memorize it. I have no clue what bar domains are, honestly. I just know that they interact with the hydrophilic heads and then they cause the bending to occur. So I have no clue. So next thing is moving on to transport. This should be like pretty much review if you guys took physio and all these other classes. So membrane and Bio 200. So membrane solid transport. So we're going to talk about the 6 different types. There's simple diffusion, where that's just simple like gaseous exchange, like you can freely cross the plasma membrane. There's facilitated diffusion for larger molecules or for ions that can't transfer on their own, so they need a protein to help, and that's what facilitated diffusion is. It doesn't use ATP, just the protein. It naturally goes down its electrochemical gradient. And then primary active transport is where we're pushing the molecule against its concentration gradient, and we're using ATP to power that, to chemically couple this transport. And then the next thing is called secondary active transport, where one of the molecules goes down its concentration gradient, and that provides enough energy for the other one to go up its concentration gradient. So instead of using ATP, we just use the natural energy from transporting from high concentration to low concentration in this case. So like if we're moving the ion from high to low, that's going to drive the substrate out to substrate in, going inwards. And then ion channel simply just allows, ions to freely diffuse through with the help of this ion channel, and basically just forms favorable interactions that pull the ion through. Obviously the ion can't go through the membrane itself. And then the ionophore mediated ion transport is basically almost like endocytosis. You could think of it, basically an ionophore will capture an ion in the extracellular matrix, and then it's able to now cross the plasma membrane, and then the ion can be released inside. So this is like more of a niche thing, but that's just basically what it is. It's almost like endocytosis in a way. And then let's talk about the transporter system. So remember I talked about facilitated diffusion and also primary active transport and secondary active transport. These use proteins that have certain motifs, general motifs. So the first one is a uniport. It transports one molecule down at a time. There's a symport where it transfers 2 molecules at the same time in the same direction. And then antiport where it transports molecules in the opposite directions. And symport and antiport are known as co-transports because they obviously transport 2 molecules or more at a time. So an example of facilitated uniport are the glucose and GLUT1 in erythrocytes. So essentially glucose, it can't pass the plasma membrane by itself. So it can't just diffuse. It's going to use these glucose transporters, and it's going to allow it to basically transfer through. So basically it's open, glucose binds in, And once it's bound, then it changes conformation. Once it changes conformation, the glucose can leave. And then it resets once it leaves back to its native state. And it's usually one direction down its concentration gradient. That's usually the general rule of thumb because we're not using ATP for this unless it's stated otherwise. And yeah, these are the kinetics of glucose transport. All you need to know is that glucose shows saturability, as you can see here, and then also specificity with the glucose itself and not other molecules. And then here's an example of facilitated antiport where we have the chloride bicarbonate exchanger. It's basically this channel over here on the right of this diagram. So we know that in respiring tissues and in lungs, we have this enzyme called carbonic anhydrase, which can convert carbon dioxide into bicarbonate or bicarbonate back to carbon dioxide. It's a reversible enzyme. And when it's converted to bicarbonate, we can actually have bicarbonate leave the cell and chloride come in the cell. So one leaves, one enters. And it's electrogenic, meaning that one negative charge leaves and one negative charge enters. So the net charge does not change at all. And it's the same thing here, but in the opposite direction where the bicarbonate comes in and then the chloride leaves out. And this is for in lung tissues. Yeah. And then here is this thing called the Circa pump. Circa pump is the smooth endoplasmic reticulum calcium pump. So what that means is in the smooth ER, or sarcoplasmic, sorry. Smooth endoplasmic reticulum. Yeah, so in the smooth endoplasm reticulum in the muscle cells, calcium is stored inside of it, so it could be used for, I think it's called smooth. But anyways, the calcium is stored in this compartment in this organelle, and the way it's forced back into that storage is the Circa pump. And so it's a movement of calcium ions. The ATP binds to and you do have to know this mechanism, the ATP binds to the end domain. So as you can see here, ATP binds to the end domain. And then from there, the end domain moves closer to the P, as you can see with this red arrow. The phosphoryl group transfer from the end domain to the P domain, as you can see here. It's going from N to P, from the green to yellow. And then once the P domain has been phosphorylated, calcium ions exit the channel and is transported across the membrane. So while this is happening, the ATP stuff and transfer, the calcium is already going into the channel. And then once the phosphate group transfers onto the P, it's going to do some conformation change and the calcium can go through. And the A domain moves to release that ADP, and now we reset and go back to baseline. That's basically the whole mechanism of the Circa pump. Primary active transport, this is sodium, potassium, ATPase. You guys should be very familiar with this already. So just give me some context. We're talking about a neuron. It's a salty banana, meaning that salty on the outside, and the banana, we know that we take it for high potassium. We eat it for the potassium. So the inside is going to have high potassium. The outside is going to have high sodium. And then the outside has low potassium concentration. The inside has low sodium concentration. So just remember salty banana, and that should pretty much stick. And remember, this is a primary active transport, meaning that both these things are going to go against their concentration gradient. That's why we need ATP in the first place. So the sodium, if it's going against concentration gradient, we know that sodium is high on the outside. So if we're putting more sodium, or if we want to go into concentration gradient, we want to move sodium from the inside to the outside. And then same thing with potassium. It's going to move from the outside to the inside against its concentration gradient. That's the easiest way to memorize. But here's like a little mnemonic as well. Na plus has 3 characters, the N, A, and the plus. So it does out. Out is 3 letters. And coincidentally, 3 sodium ions get pumped out. And then same thing with potassium, has 2 characters. And then so does N, has 2 letters as well. And coincidentally, 2 potassium ions get pumped in. Another example of primary active transport is ATP-driven proton pumps, right? The protons themselves are going down their concentration gradient, but remember, we know it can go in the reverse direction, and it does so with ATP hydrolysis. We know ATP synthase can undergo the reverse direction where it allows ATP to be hydrolyzed and to push protons against its concentration gradient again. And this is the different types of ATP synthase. There's the V type, which creates a low pH environment and does not create ATP like the one we use for ATP. It's this one right here. And then F-type, ATP synthase in the direction of ATP formation, and this one does create ATP. So something to know in terms of where protons move, directionality, and yeah. Here's an example of secondary active transport, where this is glucose in intestinal cells in your GI and your stomach cells. So essentially in this case, we're looking at the sodium glucose symporter. So as you can see right here in this blue diagram, this is the sodium glucose symporter, meaning that sodium and glucose are going to move in the same direction, and they're going to move into the epithelial cell from the intestinal lumen. We know that the epithelial cell, the conditions are high glucose and low sodium. And this makes sense. If we talk about like salty banana, for example, salty meaning the outsides are going to have high sodium, and then the inside is going to have low sodium itself. And then we know that glucose is going to be inversely related. So glucose is going to be low on the intestinal lumen, and it's going to be low in the blood in comparison to the cell, which makes sense. The cells are the ones that conduct cellular respiration. They're going to want a high level of glucose inside of it. Same with like neurons. They want to have high glucose levels inside, not on the outside. It doesn't make sense to have a lot of glucose in the blood. We want them in the cells. So hopefully that makes sense. I know I just spoke a lot, but this is just an example of secondary active transport. The glucose is going to go down against its concentration gradient, but the thing that powers it so that it can do this is the sodium ions going down its concentration gradient. And something I want to emphasize is that 2 of the sodiums are going to go through this symporter. So the secondary active transport. No, symporter, yeah. But the symporter is using 3 molecules, and it's pushing 3 through. So I just want to let you know. And then I wanted to talk about electrogenic, electroneutral. So when we say electroneutral, we're talking about just like a chemical potential gradient, meaning that in this left diagram, we're only concerned with concentration gradients. This is very easy. This we can understand intuitively. If we have high concentration to low concentration, we're going to shift it so that it equilibrates on both sides, that the water is going to shift, right? But electrochemical is a little bit more complex. We still consider the number of ions. So like the number of negative is more concentrated here. The negative is less. The positive is very concentrated here, but it's less here. However, we have to consider the positive and negative charges as well, because we know that positive and negative are going to attract each other, or positive repel, negative repel. So we have to take into consideration the electrostatic properties, the electric potential of each ion respectively, and that's the Nernst potential. That's why we do the 61.5 divided by Z log of the concentration outside divided by concentration inside, if that's the right way you do it. But point is like this accounts for the fact that these ions also repel each other if it's like and like charges. And then lipoproteins are essentially just lipid carriers. You should know these 4 types, just know the names: chylomicrons, VLDL, LDL, HDL. We know LDL and HDL basically deal with cholesterol, as you can see here. And then chylomicrons and VLDL deal with triacylglycerols, right? And so it transports cholesterol ethers, tags, phospholipids, all the lipids in the blood is going to be transported by this. And then recall that lipids aren't soluble in hydrophilic fluids like blood plasma. So they're not going to just be in the blood itself like the lipids. What's going to end up happening is they bind onto these lipoproteins and they get on like the inside essentially, and then now they're able to be transported throughout the blood. And then, here's another example of the lipoproteins in the bloodstream. This is just a very complex diagram. All I'm showing you is just that with these green circles right here, we have HDL, LDL, VLDL, and then chylomicrons. And it allows for us to transport them across the body through the blood. And and then here's descriptions of them. I basically just harped on this, but you can look at this slide more in depth. And then last slide is lipoproteins, LDL, and cholesterol uptake. So endocytosis through a receptor. So LDL receptors are LDL receptors head to the membrane. So as you can see, these are LDL receptors, and an LDL particle is going to bind to them, and it's going to cause almost like this conformational change to form a divot inside. When it forms this divot, what ends up happening is it's going to now basically endocytose it, and we see that the LDL is going to go inside, and from here it's going to be put through a lysosome and break down the lipoprotein, but then it's also going to free out the inside contents, right? We know that LDL is going to have cholesterol ethers, all this stuff. So we can see here triacylglycerols, cholesterol, amino acids, like it has this, but the main one that we're really concerned with is cholesterol. So I know I spoke a lot. All this is saying is that these particles get endocytosed inside the cell and then broken down by lysosomes. Essentially it. And that's it. Oh, and the reason why it's amino acids is because the lipoprotein itself is like an amino acid. It's like it has proteins inside of it. So when you break it down, it's going to break down into amino acids. So I know I spoke really fast, but hopefully that clarifies lectures 13, 14. I will move on to 15 and 16 right now.